Welcome friends to a new session on genetics. In this session, we will discuss extensions to Mendelian genetics, modified dihybrid phenotypic ratios due to gene interaction. First, let us see about the essential role of gene interactions in the genetic systems. According to Mendel's experiments, a pair of genes controls each character, but later discoveries proved that the interaction of more than one pair of genes controls a single character. This is known as interaction of genes or factor hypothesis. The interaction of genes was proposed by William Bateson and Reginald Crundell-Punnett, who is the creator of the Punnett square. The factor hypothesis states that certain phenotypic characters are controlled by the interaction of two or more genes. A gene interaction is the interaction of genes, either alleles or non-alleles, on a specific phenotypic character. The interaction of genes that determine the phenotype of an organism can be identified by genetic analysis. Modified hybrid ratios are the outcome of these types of gene interactions. The gene interaction occurs in between the genes located on the same chromosome or different chromosome is known as intergenic or non-allelic gene interaction. For example, epistasis, fruit color in summer squashes comes under this. The gene interaction occurs in between the two alleles of a gene called intragenic or allelic gene interaction. Example, incomplete dominance. Flower color in snapdragon comes under this. The key diagnostic is that these interactions produce modified dihybrid ratios. There are various types of interactions, and these interactions leads to a different range of phenotypes. Epistasis is a familiar intergenic or non-allelic gene interaction. Let us now examine about epistasis. Today, scientists realize that Mendel's predictions about the inheritance pattern depended on the genes he chose in his experiments. Mendel carefully selected 7 unlinked genes that control 7 different traits. According to Mendel's predictions, more than one gene controls majority of phenotypes, unlike the phenotypes that Mendel considered. Most of the organisms have more complexity than the characteristics that Mendel studied. Epistasis is one source of this complexity and can occur in different ways, resulting in a variety of different phenotypic ratios. Epistasis is a Greek term meaning above standing or stopping or suppression. Epi means above and stasis means standing. Epistasis describes the state in which a gene masks the phenotypic expression of another. In other words, epistasis is the phenomenon of suppressing or masking the expression of gene by another non-allelic gene. In some cases, a gene at one locus on a chromosome masks or suppresses the expression of gene at another locus and is known as inhibiting genes. These genes inhibit the expression of other genes. Epistatic gene is the gene which suppresses the expression of a non-allelic gene. The gene or locus which is suppressed by the presence of non-allelic gene is termed as hypostatic gene. Epistasis interactions arise because these two genes encode proteins that play a sequence role in biochemical pathway. There are two types of epistasis, dominant and recessive epistasis. If a recessive gene masks or suppresses the expression of a dominant non-allele gene, then it is known as recessive epistasis, while dominant gene masks or suppresses the expression of another non-allelic dominant gene, it is known as dominant epistasis. Now, let us look into dominant epistasis. In dominant epistasis, the dominant gene at one locus suppresses or masks the expression of another gene at a different locus, no matter of its allelic condition, whether it is dominant or recessive. The F2 phenotypic ratio is modified into 12:3:1. For instance, the dominant allele capital A of one gene masks the activity of alleles of another gene capital B and expresses itself phenotypically, then the A gene is said to be epistatic to the gene capital B. That means, the dominant allele capital A can express itself only in the presence of either capital B or small b allele. The alleles of capital B gene will be able to express it themselves phenotypically only when capital A gene may contain two recessive alleles small a small a. Therefore, the genotype capital A capital B or capital A capital B small b and capital A small b or capital A small b produce the same phenotype, whereas the genotype small a capital B or capital A capital B small b and A and B all in small produce two additional phenotypes. In this particular table, the mode of action of dominant epistasis is given. In the first column, you can see epistatic alleles. In the second column, it is hypostatic alleles, and in the third column, it is the phenotypic expression of the alleles. If you examine the first row, you can see the epistatic alleles are capital A or capital A small a. The hypostatic alleles are capital B, capital B small b, and the third condition is small b, and the phenotypic expression of alleles for these cases are capital A. In the second row, the epistatic alleles are capital A small a, the hypostatic alleles are capital B and small b. The phenotypic expression of the alleles is capital B. And in the third row, you can see the epistatic alleles are capital A small a and the hypostatic alleles are capital B small b, and the phenotypic expression of alleles in this case is capital B. Now, let us examine about the fruit color in summer squash. The botanical name is Cucurbita peppo. Squash is the common name used for 4 species in the genus Cucurbita of the gourd family Cucurbitaceae. Summer squash are a subset of squashes originated in the Americas. It is edible when the rind is tender. Nearly all summer squashes are varieties of Cucurbita peppo, which can vary considerably in color, shape and size. Summer squashes are tendril bearing plants characterized by unisexual flowers, hairy stems and a fleshy fruit of false berry with a leathery rind called a peppo. In North America, the squash are grouped into winter squash or summer squash as well as autumn squash or otherwise known as cheese squash, depending on whether they are harvested as mature fruits or immature fruits. There are 3 types of fruit color in summer squash: yellow, green and white. White color is dominant over other two colors and yellow is dominant over green. Yellow color is due to the dominant epistatic gene which is suppressed by its recessive allele W. The color of the fruit is green when the hypostatic gene is also recessive G. In this table, the phenotypes and genotypes of different varieties of summer squashes are given. In the first column, it is the genotype. In the second column, the phenotype is given. If you examine the first row, the genotypic constitution is capital W, small w, capital G, small g. The phenotype is white. In the second row, the genotype is capital W, small g, small g. The phenotype is white. In the third row, the genotype is capital W, capital G, small g, and the phenotype is yellow. In the fourth row, the genotype is small w, small g, and the phenotype is green. When a white colored summer squash variety wherein the genotype is capital W, capital G was crossed with a green colored summer squash variety wherein the genotype is small w, small g, all the F1 progenies are white color and the genotype is capital W, capital G, small g. The F1 plants were self pollinated among themselves. The offsprings resulted in the ratio 12 white, 3 yellow and 1 green in F2. The significance of this dominant epistasis is that the modification of Mendelian dihybrid ratio from 9:3:3:1 to 12:3:1 happens. In summer squash, white colored fruits are produced by a dominant epistatic allele capital W. Another dominant epistatic allele capital Y at another locus produced yellow summer squash and its allele small y for green fruits. Sometimes in this case several genes contribute to the expression of a phenotype as part of a biochemical pathway. According to this pathway, the pigment which is yellow is produced in two steps. The white color of the fruit is due to compound A. The compound A is converted to compound B by the action of enzyme 1. This enzyme is produced only when the plants have a genotype small w. Whenever capital W comes in, the necessary enzyme is not produced. On production of this enzyme, the compound A is converted to compound B and further the compound B is converted to compound C. The compound B results in green color for the fruit. If the compound B has to get converted to compound C, the enzyme 2 has to play a role, and for the production of this, the plant should have a genotype capital G. If the genetic composition is small g, the enzyme 2 will not be produced. As a result, the compound C will not be synthesized from compound B. Now, let us see about recessive epistasis. In recessive epistasis, the recessive alleles of one gene locus small a mask the action or phenotypic expression of alleles of another gene locus, wherein the genotype can be capital B, capital B small b or BB alleles. The alleles of capital B locus express themselves only when epistatic locus has the dominant alleles capital A or capital A small a. Due to recessive epistasis, the 9:3:3:1 phenotypic ratio becomes modified into 9:3:4. In this particular table, you can see the recessive epistasis and their mode of action. In the first column, the epistatic alleles are provided. In the second column, hypostatic alleles are given, and in the third column, it is the phenotypic expression of allele. In the first row, the epistatic alleles is capital A small a, the hypostatic alleles could be capital B, capital B, capital B small b, small b, small b. Then the phenotypic expression of the allele would be capital A. And in the second row, the epistatic alleles is capital A, capital A, capital A small a. The hypostatic alleles could be capital B, capital B, capital B small b. The phenotypic expression of the allele is capital B. And in the third row, you can see the epistatic alleles can be either capital A, capital A, capital A small a, and the hypostatic alleles capital B small b. The phenotypic expression of allele is capital B. Recessive epistasis in flower color in blue-eyed Mary. The botanical name is Collinsia parviflora. It is a species of flowering plant in the family Plantaginaceae, known by the common names small flowered Collinsia and maiden blue-eyed Mary. When a homozygous white flowered plant was crossed with a homozygous magenta flowered plant in F1, all blue flower colored plants were produced. When the F1 blue flowered plants were crossed among themselves, they produced offsprings in F2 generation as blue, white and magenta flowers in the ratio of 9:4:3. In this table, you can see the recessive epistasis and their mode of action for flower in blue-eyed Mary. In the first column, it is the epistatic alleles are given, second column represents hypostatic alleles, and the third column represents the phenotypic expression, and finally the F2 phenotypic expression in the fourth column. In the first row, the epistatic alleles is capital W small w and the hypostatic alleles are capital M, capital M, capital M small m, small m, small m. In this case, the phenotypic expression of allele is capital W, and the F2 phenotypic ratio is white, and there are 4 progenies. In the second row, the epistatic alleles are capital W, capital W, capital W small w. The hypostatic alleles are capital M, capital M, capital M small m. The phenotypic expression is capital M, and in the F2 the phenotypic ratio is 3 and it is represented by magenta. In the third row, the epistatic alleles are capital W, capital W, capital W small w. The hypostatic allele is capital M small m. Here the phenotypic expression is small m, and in the F2 the phenotypic ratio is 9 represented by blue color. Here complementation results in a wild type F1 heterozygous blue represented by capital W small w, capital M small m. However, in the F2 a modified 9:3:4 phenotypic ratio is produced instead of 9:3:3:1. In this example, the small w allele is epistatic on capital M and small m. On the other hand, capital M and small m can be expressed only in the presence of the allele capital W. In this experiment, the recessive allele is epistatic. This is the case of recessive epistasis. If one or more intermediates in a biochemical pathway are colored, then a different F2 phenotypic ratio is produced. In this example, the enzymatic pathway for the color production from the plant blue-eyed Mary is shown in the figure given. When the blue flowered plants are selfed, the ratio produced is 9:3:3:1, of which 9 offsprings are blue colored. This is because the both enzymes are active in this case. The genotypic composition will have W plus and M plus. Therefore, both enzymes are produced over here, which will finally result in blue coloration of the flowers. In the second row, 3 flowers are magenta colored. Here the genetic constitution is W plus and M plus is absent over here. As a result, enzyme 1 alone is produced, which will culminate in the production of magenta color alone. And in the third case, 3 white flowered plants are produced, wherein the genetic composition is M plus. Therefore, the enzyme 2 is synthesized over here and there is no production of flower color and the flower remains as white. And in the last case, only a single white flower is produced, and here no enzymes are produced because W plus as well as M plus are absent. As enzymes are absent, no substrate conversion takes place and the flower color is white. Now, let us look into other types of epistatic interactions. One is duplicative recessive epistasis. Duplicate recessive epistasis is the complete dominance at both gene pairs. However, when the either gene is homozygous recessive, it masks the expression of the other gene. For instance, in man, normal hearing is due to 2 dominant genes, capital A and capital B. But the recessive alleles small a and small b of the 2 dominant alleles are epistatic when they are homozygous. The paired recessive allele of one dominant gene masks the expression of other dominant gene. Thus, a person with the genotype small a, capital B would be a deaf mute because the genes small a are epistatic to the gene capital B for normal hearing. The paired recessive gene small b is epistatic to the gene capital A, which is responsible for normal hearing. This is a case of duplicate recessive epistasis where either of the 2 recessive genes may be epistatic to the dominant alleles of the other. When a normal hearing person represented by capital A, capital B is crossed with a deaf mute represented with small a, small b, it produces a normal hearing person with the genotype capital A, capital B, small b in F1. If the F1 individuals are crossed themselves, they produce 9 normal persons and 7 deaf mute persons. Now, let us see about duplicate dominant epistasis. Complete dominance at both gene pairs, however, when the either gene is dominant it masks the expression of the other gene. Independently, the ratio will be 15:1 if a dominant allele of both gene loci produces the same phenotype without cumulative effect. Complete dominance at both gene pairs, but the either gene when dominant epistatic to the other. For instance, in seed capsule of shepherd's purse, gene pair capital A, triangular shape is dominant over ovoid gene pair capital B, and the triangular shape dominant over ovoid produce F2 offsprings in the ratio 15 triangular 1 ovoid. Now, let us see about the biochemical basis of epistasis. Every cellular chemical reaction involves the stepwise conversion of one initial substance called precursor to another called end product. All the subsequent steps of a chemical reaction constitute the biosynthetic pathway, which includes various steps, and each step is catalyzed by a specific enzyme. Each specific enzyme is synthesized by a specific gene. Number of enzymes are involved in the conversion of one material to another material. In other words, series of genes are involved in the conversion of one material to another material. The simplest example to explain biochemical basis of epistasis is albinism. It develops due to the lack of melanin pigment, which produces black color. Melanin synthesis is not one step reaction like all other cellular chemical reactions. Rather, it is a 4-step process with each step mediated by a specific enzyme. These enzymes, which catalyze the reaction at every step, are produced by some specific dominant gene. We can get the idea of process with the help of the figure. The precursor is converted into the metabolite 2 only by the action of enzyme 1, and for the production of enzyme 1, the genotypic composition should be either capital A or capital A small a. The conversion of metabolite 2 to metabolite 3 occurs with the help of enzyme 2. The enzyme 2 is synthesized only when the genotype is capital B or capital B small b. The conversion of metabolite 3 to metabolite 4 happens with the help of enzyme 3, and the genetic composition required for the production of enzyme 3 is capital C or capital C small c. The metabolite 4 is finally culminates in the production of melanin, which happens with the help of enzyme 4, which is synthesized only when the genotype is capital D or capital D small d. As the gene capital C undergoes mutation, mutated gene small c could not produce enzyme 3. No production of further metabolites happens, and the result is albino. This is illustrated in the figure given in the screen. Now, let us examine about duplicate genes with a cumulative effect. This could be additive genes or polymeric genes. Two independent dominant genes, whether present in heterozygous or homozygous condition, express similar phenotypic effect when present individually, but produce a double effect or a cumulative effect when found together. In other words, two completely dominant genes controlling a character produce identical phenotypes when they are alone, but both genes are present together. Their phenotypic expression is enhanced as the effect of the two genes were cumulative additive. In additive gene action, each dominant allele of the two genes produces equal and identical effect on the character. The effects of the all dominant allele at the two loci are additive, that is, if the dominant condition at either locus produces the same phenotype, a 9:6:1 modified dihybrid ratio is obtained in the F2 generation as phenotypes produced by single gene dominance of the two different genes are similar. In this given table, you can see the mode of action of duplicate genes with cumulative effect. The first column represents epistatic alleles, second hypostatic alleles, and third phenotypic expression of alleles. In the first row, the epistatic alleles is capital A small a, hypostatic alleles capital B small b. The phenotypic expression is neither small a or small b. In the second row, the epistatic alleles is capital A small a, the hypostatic alleles is capital B or capital B small b. The phenotypic expression of alleles is capital B only. In the third row, the epistatic alleles is capital A small a. The hypostatic alleles is capital B small b. The phenotypic expression is capital A only. In the fourth row, the epistatic alleles is capital A small a. The hypostatic alleles is capital B small b. The phenotypic expression is capital A and capital B. Another example would be fruit shape in summer squash, Cucurbita pepo. Certain species of summer squash can exist in different shapes. They are long, spherical and disc shaped. In case of Cucurbita pepo, the fruit shape is controlled by two genes, gene capital A and gene capital B. The disc shaped summer squash has both the dominant allele capital A and capital B with a genotype capital A either with capital A or small a, capital B either with capital B or small b. Elongated summer squash produced when both genes are in recessive combination small a, small b. Spherical summer squash has either of the two dominant genes capital A or capital B with a genotype capital A could be with capital A or small a, capital B, or small a, capital B with capital B or small b. When two different races of sphere shaped summer squashes with genotypes capital A, small b and small a, capital B are crossed, the F1 progenies are all disc shaped with the genetic constitution capital A, capital B, small b. When these F1 progenies are self pollinated, interaction between both the dominant genes give new phenotypes in the ratio 9:6:1, wherein 9 disc shaped are formed, 6 sphere shaped are formed and 1 elongated shaped. Epistasis or interactions between genes has long been recognized as fundamentally important to understanding the structure and function of genetic pathways and the evolutionary dynamics of complex genetic systems. With the advent of high throughput functional genomics and the emergence of systems approaches to biology as well as a newfound ability to pursue the genetic basis of evolution down to specific molecular changes, there is a renewed appreciation both for the importance of studying gene interactions. Epistasis was originally a concept from genetics, but is now used in biochemistry, population genetics, evolutionary biology and computational biology. Now, let us summarize the foregoing discussion of gene interactions. Genetic analysis can determine that the gene interactions determine the phenotype of an organism. A gene can express itself in the homozygous state or heterozygous state as dominant or recessive conditions. According to Mendel's experiments, a pair of genes controls each character, but later discoveries proved that a single character is controlled by more than one pair of genes, known as interaction of genes or factor hypothesis. Gene interaction is of two types, allelic and non-allelic gene interaction. Allelic gene interaction occurs between the two alleles of the same gene, but non-allelic gene interaction occurs between the alleles of different genes. Epistasis is a familiar intergenic or non-allelic gene interaction, the phenomena of suppressing or masking the expression of gene by another non-allelic gene. Epistatic gene is the gene which suppresses the expression of a non-allelic gene. The gene or locus which is suppressed by the presence of non-allelic gene is termed as hypostatic gene. There are two types of epistasis, dominant epistasis and recessive epistasis. In dominant epistasis, the dominant gene at one locus suppresses the expression of another gene at a different locus, no matter of its allelic condition, whether it is dominant or recessive. The fruit color in summer squash is an example, and F2 phenotypic ratio is modified into 12:3:1. Sometimes the recessive alleles of one gene locus mask the phenotypic expression of alleles of another gene locus is known as recessive epistasis. Flower color in blue-eyed Mary is an example of recessive epistasis. In this, the F2 phenotypic ratio is modified into 9:4:3. Duplicate recessive epistasis is the complete dominance at both gene pairs. However, when the either gene is homozygous recessive, it masks the effect of other gene. Duplicate dominant epistasis is the complete dominance at both gene pairs. However, when the either gene is dominant, it masks the effects of the other gene. In cumulative effect of duplicate genes, two independent dominant genes, whether present in heterozygous or homozygous condition, express similar phenotypic effect when present individually, but produce a double effect or a cumulative effect when found together and modified dihybrid ratio of 9:6:1 in the F2 generation. Fruit shape in summer squash is an excellent example of cumulative effect of duplicative genes. Now, try to work out some of the given assignments over here. 1. How do we relate epistasis to complementation? 2. Explain how epistatic gene interactions produce a range of different phenotypes. 3. Make a cross between two homozygous white flowered and magenta flowered blue eyed Mary up to F2 generation. 4. Explain the biochemical basis of epistasis. 5. Explain duplicate dominant and duplicate recessive epistasis. Now, let us see some of the references with regard to the discussion we had today. 1. Genetics, a molecular approach plus mastering genetics with e-text, 3rd edition, authored by Peter in the year 2011. 2. A book with the title An Introduction to Genetic Analysis, 7th edition, published from New York. The authors are Griffiths, Miller, Suzuki, Levallontine and Gilbert, published in the year 2000. A third publication which you can take as reference is Principles of Genetics, 8th edition, authored by Gardner, Simmons and Nestor in the year 2006, published by John Wiley, New York. A fourth reference would be entitled Concepts of Genetics, 10th edition, authored by Klug and Cummings in the year 2011. A fifth reference is entitled Principles of Genetics, authored by Gardner and Nestor in the year 1984 and published by John Wiley, New York. A sixth reference is entitled Principles of Genetics, authored by Sinnott, Dunn and Dobzhansky in the year 1958, published by McGraw-Hill, New York. Now, some of the websites which you can refer to for further reading is www.genetics.org. Second one is theagricos.com. Thank you for watching this session. See you next time. Until then, bye.